So this is your first corrective cell division. And let me just see if I can make some notes on here for you. Process A. Actually, process A versus process B. Okay, so it says, what's the purpose? This is process A right here. This is A, right? So it says, what is the purpose of process A? You said to make sex cells or sperm and eggs. So if we look at our product. This is our start for both of them. And these are identical, right? That's identical to that. They both have 4 chromosomes, right? But this is our product, right? And the product here is the same here. It goes from 4 chromosomes to 4 chromosomes. But the product here is kind of half, right? Do you see how these are half? And what is half? What's the word for half when we'll talk about chromosomes? It's called haploid. So haploid means that there's no pairs of chromosomes. So do you see how there's, if I was to draw these chromosomes, I would draw the original chromosomes like this. I draw one big blue one, like this, and one small blue one, like this. And then I would draw one big red one, like this. And I would draw one small red one, like that. So the reason I'm showing you that is because this is pairs, right? There are 2 pairs. And if you have pairs, we call that diploid. Diploid means doubles. Diploid double. Okay? But in the end, you can see here that it's either red, maybe there's a big red one, and maybe there's a, where'd the blue go? I don't see any blue. Is that blue? I guess that's blue. And maybe there's a small blue one, and those are no pairs. And if it has no pairs, it's half, and that is haploid, haploid half. So when you look at this, these are haploid and these are diploid, right? Diploid, because they have pairs. Okay, so you said process A was meiosis, and that makes sex cells or sperm and eggs. Now, aren't sperm and eggs, isn't sex cells? You said they're sex cells, right? Aren't sex cells half? Aren't they haploid? Because they're half, right? So, yeah, do you see what I'm saying? Process A makes diploid. You said, what's the product of process A? The result of meiosis is haploid daughter cells with chromosomal combination. Haploid daughter cells with half the chromosomes. Half the chromosomes. Why? Because we are making haploid, because we want to make sex cells, and sex cells have to be half, right? Got to be half. Because when the sperm comes into the egg, right, in a human, this would be 23, and the egg would also be 23. And when you fuse them together, what do you get? 23 plus 23 equals 46. And so the baby, which would be the baby, right? And the baby is diploid. The only cells that are haploid, that I don't like the diploid. The only cells that are haploid are sex cells, sperm, egg, pollen, etc. Right? So process B, you said mitosis. Well, maybe you thought it was mitosis because there's more going on. No, this is trying to split in half, right? Also, right here, they're doing something called crossing over, which is mixing it up. So you want to mix it up and you want to split it in half. So that's meiosis. So your process B and your process A, maybe you flip those. What is the purpose of meiosis? For growth and replacing worn out cells. That's true. Check. Let's see. What's the purpose of process A? Yeah, you flipped them, right? Okay. What is the product of process B? Mitosis is a single process where a single cell divides into identical daughter cells. And yeah, so you're dividing into daughter cells. And you're dividing into daughter cells because you want to grow and you want to replace lost cells. But the problem is that your processes are, kind of flipped for most of these. You want to switch them. Use the word bank concept to explain, describe, contrast these processes. So basically, mitosis is the process of producing identical body cells for the purpose of growth or to repair or replace lost cells. Those are going to be diploid. Meiosis is the process of making haploid sex cells like sperm and egg. This happens inside the testes or ovaries. Crossing over helps to make more variation. And so these are all correctly associated with meiosis, but it'd be nice to put them into a sentence if you can. Let's see, mitosis, you said body growth, repair, and replacement lost cells. No crossing over, good. And this is also diploid, right? If you remember that. So yeah, you haven't done badly on this because a lot of stuff is right. A lot of stuff is just flipped. So use this to see if you can fix your cell division, and hopefully that helps. I'm going to go on to the second one, which is the DNA exam. So looking here, what we have is we have this little thing right here, this little dotted area, and this is a monomer, and it's the monomer that makes up the DNA ladder. And so, for instance, we have the DNA ladder, and it looks a little bit like this when you put it together, right? And there's slightly different shapes, right? Like this, right? And these monomers are called nucleotides. How many monomers do you see there? Hopefully you see 4 nucleotides. Okay. How many monomers do you see here? There's a lot. How many monomers do you see inside here? There's more than 4. All right. Hopefully you can see there how many monomers are inside there. Okay. So this process, the whole process is called protein synthesis. And we're making proteins. And the first part of protein synthesis is transcription, which means to rewrite. So it's called transcription. So transcription is the process of creating mRNA. This is mRNA. Process of creating mRNA out of DNA. And this is going to make what's called a gene. And a gene is going to let you do protein synthesis. Okay. After transcription, then we do the translation of the gene. And so here we're going to translate, we're going to do translation, and we're going to do translation of the mRNA gene into a protein. And what are proteins made of? Well, proteins are made of amino acid. Made of amino acids. Okay, so where's the protein? Well, the protein is this whole thing right here. The protein is this whole thing right here. That's that molecule. Now, what are the individual molecules? Maybe I'll put in a different color. I'll put in green, right? So the amino acids are going to be the individual molecules, right? And so actually, there's 5 amino acids here. You might think, well, aren't there 6, Mr. Morrison? What about this one? Well, do you see that this one is actually shaped like a stop sign? And the reason it's shaped like a stop sign is because that one is called a that one is called a stop codon, UAG. It's called a stop codon, and a stop codon tells this is the end, right? This says this is the end. You need to stop here. It's called a stop codon. So that's 1, 2, 3, 4 amino acids plus a stop codon. This is the start codon, AUG. AUG is the start codon. And so it says, hey, start here and then read this way and finish here. So how do we read it? Well, we have these letters, right? AUG, AUG. And the second letters, we got to look up here. Second letters are U, U, G. U, U, G. Okay. Translate into amino acids. These three-letter things are amino acids. These are mRNA. mRNA. So A. Let's do the first one. I'll do it in red. A, U, AU gives me this right here. It's going to be one of these four. So now I look over here. A, U, G. Not this G, not that G, not that G, this G, because it's this intersection. It intersects right there, and then finally there. So that is met start. That's methionine, and that's always the start codon. And if I go up here, this one is going to be methionine, and this is the start codon. Okay, I'll do the next one in blue. So this one right here, I'll do in blue, this whole section. I'll do this in blue. U, U, G. U, U, G. Okay, U, U, G. I'll do a little bit darker so you can see a little bit better maybe. So U is the first letter. Second letter is U again. So these two meet here. Well, which one? It could be either Phi with an A or Lu with a B. So U, U, G. G is this one. So I go over, it must be this one here, Lu with the B. So I write leucine, Leu is leucine, that's an amino acid. And then I write the letter B. And then you just continue on with that. So hopefully you can get that. Oh, the T-shaped molecules. T-shaped molecules, trigonal planar, no. T-shaped molecules are bringing, they're actually transferring. They're actually transferring. So they got three things sticking off the end, and they're transferring something special. They're transferring these little amino acids. And so a tRNA stands for transfer RNA, and their job is to transfer amino acids. And so if you can think of them as like little workers with a little wheelbarrow, they're going to bring their little wheelbarrow in here, and they're going to read the code and build the protein out of amino acids. That's the job. The code is made of mRNA. This one is mRNA. The code is read in the process of translation. The code is read by tRNA molecules. The tRNA molecules know exactly which amino acid to bring. They don't need to read the code. They know the code by heart. And we know the code, though. We know that each tRNA will bring an amino acid. Which amino acid will it bring? It will bring the correct amino acid according to the code. And that is an mRNA code. Those are called codons. So mRNA has the codons, and tRNA has what are called anticodons. You don't really need to know that, I don't think, on this test, but they're called anticodons. They are the matching codons to the anticodons. Anticodons, codons, anticodons, and so they will bring the correct amino acid to build the protein. So I think that answered everything we got here, pretty much. Let me know if your problem. This is complementary base pairs here. What does G go with? What does C go with? What does A go with? And what does T go with? Right? What's this process called? What's this process called? Right? The purpose of this process, transcription, is to do what? So hopefully you got that out of these videos. Watch them a couple times if you have to. See if you can answer them, and then shoot me an email with any questions that you might have along the way.